So now we're going to talk about inheritance of genetic diseases, and this is the first session of this section. And so first of all, these are the objectives, which we're going to go back to at the end, and hopefully by the end of the session, all of these words and concepts will be very clear. And starting with some important definitions. So unfortunately, this part of genetics has a lot of jargon, and we need to kind of decipher it in order to properly understand these conditions. So first of all, going back to the previous session, what is a gene? A gene, just to refresh your memory, is just a distinct sequence of DNA forming part of a chromosome, and the order of which, so the order of the nucleotides, the DNA, determines then an amino acid sequence of a protein. And another very important term is the locus. As you can see here, this is an example of a made-up chromosome. The locus is simply the location of a gene on a chromosome. All that refers to is just the position on a specific chromosome. Like, let's say a gene X. Gene X is on chromosome 2, position 3,000,002, something like that, whatever. And another important concept, which is linked to the idea of gene, is alleles. So first of all, does anybody know what an allele is? Have you guys heard the term or are aware of its existence? Please don't feel awkward to say anything. The point of this is to learn, so it's not a bad thing to be a mistake. Well, it's fine. So just an allele is just simply can be defined as an alternative form of a gene, which sorry. Oh, apparently, Sainabou, can you hear me?
Yes, I can hear you.
And apparently some people can't unmute themselves. Do you read the group chat?
Oh yeah, that's because of what we incurred the last time. So if you want to comment, just raise your hand, and then we can unmute you. And yeah.
Okay, understood. Okay, then we can do it later for the more interactive part. Anyways,
Yeah.
so allele is simply just like an alternative form of a gene, and that is normally they differ from each other by few bases. So for example, you can have gene A and it can have different forms called alleles. And this is very important because different alleles will give rise to the diversity that we see in humans, and that can be eye color, that can be disease, height, whatsoever. And continuing on understanding what allele is, we often have alleles that can either be dominant or recessive. What that simply means is that, like, the effect they have on a phenotype. Phenotype is simply just like the characteristic of an individual. So a dominant allele is where one copy of this different allele is enough to cause a change in the context of diseases to cause disease. Instead, a recessive allele by itself, so if you only have one, nothing out of the ordinary happens. But if you have two, then you do see a difference. And an example of this is, for example, like although it's a bit simplified, but dark eyes are dominant alleles, which means that you can have only one form and you have a darker eye. And instead, something like blonde hair is instead of recessive, where you need two copies of the same altered this lighter hair allele to then cause the phenotype of having blonde hair. And so this is the concept of dominant versus recessive, which we're going to go through more in depth later. And then lastly, which is very important, the idea of a genotype and a phenotype. So genotype, as the word gene suggests, simply means the genetics of an individual. And that means what alleles, what genes, what individual genetic composition the individual has. And instead, phenotype is simply the things that you can see, the characteristics. Can be height, can be hair color, can be anything that is visible and shown. In a way, the genotype is like we said about like last lecture, is a bit of a book, and a phenotype is the effect, so what you can actually see and in concrete. So these are very important definitions, which we'll try to build on in this lesson and in this discussion. And okay, So now we're gonna draw from our previous lectures to better understand inheritance and how it's important um, in genetic diseases. So first of all, does anyone know what this diagram is called? So this diagram, it essentially it's a overlays all the chromosomes of a human. And does anybody know what this type of gram is called? You guys can write down in a group chat. That could be quite interesting. And if nobody knows what an issue, it's very a niche term. It's called a karyograph, and it just simply showcases what the chromosomes a person has, or different species, whatever. And what's very important is that, which will come a bit more important later, is that there are two kinds of chromosomes in general. We have the non-sex chromosomes, which are called autosomes, which are all the chromosomes 1 to 22. And then we have two pairs of sex chromosomes, which are the ones, as the name suggests, that make a person either male or female genetically. And they're important because the inheritance of diseases changes depending whether a mutation, a genetic a disease-causing mutation, is either on the autosomes, so on the non-sex related chromosome, or on the sex chromosomes. So this will come back later. But very, very important understanding is that we all have two copies of all our chromosomes, and we inherit one of each copy from our parent, from our dad, and one from our mother. This means that for every gene, we have two copies, one paternal, one maternal copy. And exactly. And yeah, and continuing thinking of reproduction, and so how we make so like how we have children. So we humans have, as I just said, 23 copies, like 23 two copies of the 23 chromosomes, and hence we're called diploids. Or the D stands for the two. So D is in I think it's Latin, and it means two because we have two copies of each chromosomes. Nevertheless, if you think about it, if you have a mother and a father, you can't fuse two cells that have 46 chromosomes each. Then you would have a massive cell that has 90 plus chromosomes, and that just does not make it's called a viable human. Hence, when we produce sperm and egg cells, they go through a specific type of it's called cell division called meiosis, which is a process by which the sperm cells divide and become mature, and they lose half of their chromosomal of the chromosomes. So if you compare the cardiograph of sorry, my phone is sorry. If we compare the cardiograph of a normal cell, like a cell, a neuronal cell, a skin cell, a tongue cell, an eye cell, it will have 46 chromosomes. Instead, our sperm and egg cells have half, 23, which is vital because then when the egg and sperm cells meet, then we form one single from an embryo that has 46 chromosomes. So this reducing the number of chromosomes is necessary for viable reproduction and formation of babies. And so, as highlighted here, it's a necessary step for the genetic material of the mother and father to be combined. And this is very important because then it will play on how genetic disorders are actually inherited. So, thinking of giving an example, making more concrete, let's talk about eye color. So first of all, this is an example. Actual eye color is much more complicated, but we're going to simplify it a bit to understand better genetics. And okay, let's think of an example where the gene called E controls eye color. E, capital E, is the dominant allele for dark eyes, and small e is the recessive allele for light eyes. Here, it's very important that we continue to remember the dominant and recessive. And my first question is, let's say that we have a random person called John that has a genotype two capital E's. What will their eye color be? Does anybody want to unmute and talk about it with me?
Maybe they can put it in the chat. I can try reading it out.
Oh yeah, that also works. Please, whatever is easiest for everyone. If we are not sure, honestly, I think it's nice. Like, there's no nothing happens if you get it wrong. But I think the key term is, so here we see that the not large, the dominant allele, which is the capital E, causes dark eyes, and John has two capital E's. So their eye color, what would it be? Anyone wants to have a guess? Take a guess.
Sainabou says it's brown in the chat.
Perfect. Yes, that is indeed correct. And exactly, as the person in skin, John, has two copies of the allele for dark eyes. Perfect. Good job. And continuing instead, well, let's give again another example. A woman called Mary, and she has two and non-capital, like small e's. What will instead her eyes be? Any guesses?
Someone in the comments says light, Natasha, Maria.
Exactly. Yes.
But
Good
they look
job.
good.
That is correct because we have two copies of the same allele that yield the light eyes. But now we have a third example, which is a bit more complex. And instead, with third person, Lucy, that actually has two different alleles. So she has capital E and then small e. And in this case, she technically has genetic material to produce either dark or blue eyes. But do we is there something about the genes that we know that we read about that could tell us potentially what kind of eye they would have if they were large E, small e?
Natasha says brown, dark brown because E is dominant.
Exactly. That is the key understanding that I was trying to get at. Because dominant, it means it's called one allele is enough, so the darker eyes will dominate over the recessive blue eye. Exactly. Perfect. And so here we see an example where the genotype and phenotype can be a bit mixed up because John, first person, and Lucy, third person, have the same phenotype, so the same dark eyes, but they do have a different genotype as he has two capital E's and Lucy has one capital, one small e. And this doesn't affect them as people, but it may affect generations forward. But yes, perfect. And so good job to everyone. And so let's continue. Now let's think about inheritance and how John and these people will affect their offsprings. So again, we're still continuing with the idea of the gene E, and let's add a very important key understanding is that a person has two alleles for each gene. But as we just talked about, the egg and sperm cells only can have one allele. So because we only inherit from one parent, one allele they have, because we kind of become a mixture of our parents. And so um, if so when we make sperm and egg cells, the sperm and egg cells will have a half 50-50 chance of either having either of the two alleles we have. So thinking again about John, if his genotype is two capital E's, what genotype will his sperm cells be? Obviously, it depends on which cell, but we're talking about ratios. So what can we expect? Any guesses? So just a reminder that obviously the sperm cells only have one allele, and because they're haploids, not diploids, so half the genetic content, and so they will be as likely to have one allele as the other. So any guesses? Okay, if you don't have any guesses, they're completely fine. And all sperm cells will carry the dominant E allele. This is because we have, in this case, John, that has two dominant alleles. Only has the two dominant E's, and so he can only when sperms are produced, they will always have either of the same allele if that's all he has. And continuing on this example, let's now think of Mary with its production of her egg cells. Instead, because her genotype is two small e's, what will her egg cells instead? What genotype will her egg cells be? Does anybody can take a guesses? It's quite similar to the first question, just like the opposite in a way. Sainabou, are you reading the answers? Is there any?
Yeah, there's no comments in the chat. Light eyes. Someone says light eyes.
Yes. Yes. So yes, that is correct, but I think I wrote the question wrong. Sorry, it was my fault. But light eyes is correct. And just to clarify, I was also asking about the genotype O of the X cells, and because oh, sorry, that is wrong. We have to fix that. Sorry, I fixed that very quickly. Sorry. Anyways, and because yes, so she will carry all the X cells will have the recessive small e, and that's very important to think about later. And then the third question is now looking at Lucy, who has the genotype big E small e, and not just what eye color they will be, but also her X cells. What genotype can they be? And this is more complicated because she, unlike Mary and John, has two different alleles. So her egg cells will have two different alleles as well. So any guesses on what genotype her eggs could have?
Jainaba says Mary has a homozygous recessive.
It's not homozygous. This will go for later. The answer is that because she has the two copies, so big E and small e, her X cells will have 50% chance of having the big E and 50% chance of having the small e. So what this slide is trying to show everyone is that we as humans, we have two copies of this of each allele. Not all of a gene, sorry, not always this gene would be different. So we might have, as an example of Q1, Q2, two of the same allele. But sometimes we have a context in which we have one gene, two alleles. And when we have the same allele, so question 1 and question 2, we can obviously only give to our sperm cells and our egg cells one allele. But when we have two different alleles of the same gene, there will be 50-50 chance of us either giving the small or the big allele. And so the reason why it's saying 50-50 is because when you get the production of sperm cells, the chromosomes are separated in a random way, which means that as we have two copies of each, there would be as likely chance of having one allele as the other. And so this is very important to understand how genetic diseases are inherited. And a keynote, as we said before, even the phenotype is the same. The genotype and inheritance can differ, and this is what I talked about before about John and Lucy having the same eyes, but then having a different genotype. Okay, Now we're going to translate what we have understood. So the fact that we have two copies of each gene and that we give to our offspring only one copy into the context of genetic disorders. So the first thing we're going to go through are autosomal dominant genetic disorders. And whenever we encounter a complex word in science, what is always useful is to break it down. Because if you break down a complicated science word, it oftentimes makes sense. So, we have the word autosomal and dominant. Does anyone remember what the word autosomal means? You can write in a group chat or be unmuted if you want. So, just like a small recap, if we go back
Someone
to
in the chat said somatic. Dam said somatic.
yes, yeah, exactly. So it's also like non-sex related. So if you go back to this cartograph, so all the chromosomes, we see that all the chromosomes except for X and Y are autosomal, so not linked to our sex. And this, sorry, and this is very important. Sorry, I'm lost. And this is very important because it will then change the type of inheritance that we have. And then dominant. So with autosomal, so it's not on a sex chromosome. And now with a dominant genetic disorder. So as we said before, does anyone remember what dominant means in the context of genetics? So dominant means that one allele is enough to showcase an abnormal phenotype. And so this can be applied to, as we did before, to eye color, and the O in this context, abnormal refers to disease context, to a disease context. And so as we saw before, John was two, I know, actually, no, that best example was Lucy. She was big E, small e, because the darker eye color was dominant, she had dark eyes. Okay, so now if I'm telling you that we're studying a disease that is autosomal dominant, and then John suffers from this disease, the example that I'm giving is Huntington. If Huntington is caused by the H gene, what is John's genotype? So we know that the disease is autosomal dominant, and we also know that he's suffering of this disease. Hence, what is, does anyone want to take a guess of his genotype from the information that we have? Please take a guess. Even if you're not sure, you can ask a question and we can talk about if there are any confusions. Any guesses, any ideas?
Dam says H.
Yes, that is partially correct, but so when we show the genotype of a person, we always have two letters because each letter refers to one allele, so to one form of the gene that they have. So H is correct, but it's partially correct. So we want something that is like either two big H's, two small H's, one big, one small. Do we have any guesses? So it's very
Yeah.
similar, yeah?
Someone in the chat said 2H, so that's capital H and capital H, or capital H and small h.
Exactly. That is exactly perfect. As and the reason why is we all as to reiterate, all humans have two alleles of all genes essentially, and we have in this context an autosomal dominant genetic disorder. So this means that the person that is suffering from the disease can either be two big H's or one big H, one small h. And this is because it's dominant. One big H is enough to confer the disease, to cause the disease. And we're going to continue this example. And from now on, assume that and that the genotype is big H, small h, just for future reference. Okay, now instead, let's say that Mary is a healthy woman. She does not have Huntington, but she still obviously has the gene that may cause it, but she does not have the allele that causes it. So what instead is Mary's phenotype? And remember, as humans are diploids, we always have two genes, and say two forms of every gene. Are there any guesses on Mary's sorry, I'm a genotype, not phenotype. Sorry, I will correct that.
Dam says 2 h.
Exactly. She can only have two small h's because because she's healthy. And otherwise, if it's dominant, that's the only thing she has. Exactly. Now, let's think of an example. Let's think of John and Mary's. Let's say that we are a doctor and we have John and Mary coming to our hospital, and they say, okay, John is suffering from this disease. Are we at risk of having a child that also carries the disease? And to do this, we need to understand how genes are spread from one generation to another and apply this. So a very easy way to figure this out is to create a Punnett square. So to do this, we're going to do this together. I recommend, if anybody has paper, to get some paper and write this down as we go, because it's very visual, it's very useful. I will be doing it as well. So what a Punnett square, this is called a Punnett square, and simply it's a way to think about the genetic movement, the movement of genes between a couple, between two individuals. So as we see here on under the headers, these two columns, we have parental allele A, parental allele B. What does this mean? In this context, we have John, and we know that he is big H, small h. So we have to like think about putting the big H here and small h in here. And the reason why is that we're trying to figure out all the possibilities that can occur from John's sperm cells and Mary's egg cells mixing. So let's assume big H here, small h here. And instead here, we want to put the maternal alleles. From the example that we just talked about, we know that she's healthy, and so we know that she can only have two small h's here. So the headers, so on the columns and the rows, are the paternal and maternal alleles, whereas these one here are the potential genotypes of all the possible offsprings. So there are four possibilities that would have four different possible outcomes. And we're going to figure out whether this will cause disease or not in any of their children. So what are the two? Does anyone take a guess of the paternal allele A and B? With A and B, I just mean 1 and 2. Does anyone want to take a guess what they are? So we just talked about it about here. And yes. Does anybody know what they could be? And just to give more context, the reason why we're separating the different alleles by parent is because, as we said before, when we make a sperm egg cell, we lose half of our genetic material. Any guesses on the paternal alleles that can be passed down here and here? Sainabou, any answers?
Capital H and small h.
Perfect. Exactly. And just to recap, so as you saw here, we know that his genotype is capital H, small h. And so his sperm cells can only have capital H or small h, and they will be present half the time. So half the sperm cells will be capital, and the other half will be small h. Now, talking about the mother, so she is a healthy individual. So what will the genotype of her egg cells be?
H small h
Exactly. Perfect.
from Abdullah.
Now that we know our parents' possible alleles, the next step is very easy. So if you can, please like write down. It's very it's use it helps you understand. Um, the first one here, you can see my mouse. So the first box, the first cell, we have our paternal allele H and our maternal one, small h. So we have big H, small h, because we're combining this one and this one. It's very simple. And then let's go to the next one. So now we're looking at the third row, and no, sorry, the third column, first row. So here. So what will this one be? So we're combining paternal allele B and maternal allele A.
Abdullah says small h, and so does Natasha.
Exactly. Perfect. That is perfect. That is very good. Good job. Now, going back to this one, so paternal allele A and maternal allele B. What will this one be?
Abdullah says small h and capital H.
Exactly. Exactly. The order, yeah, that's perfect. The order doesn't matter if it's yeah. And the last one, so we have paternal B, maternal B.
Small h from Abdullah.
Good job. That's very good. Okay, so now what this means is that the ratio of genotypes of our embryos of our babies from this couple will be one big small to one small small. So with an equal chance, 50-50, of having uh, one big, one small, and two small. Okay, translating this from genotype to phenotype. Does anyone want to take a guess on if you were the doctors, as you go back to the example, are John's and Mary's children at risk? So half the children will be the same genotype as their dad, big H, small h, and the other half will have the same genotype as the mother, small h, small h. So what does this mean in the context of translating genotype to phenotype? And if anybody wants to speak to ask a question or to clarify something, you can put your hand up or write down, can I speak for a second? And that is very simple.
Abdullah says for every pregnancy, there's a 50% chance of having a diseased child.
Exactly. That is perfect. That is the right answer. So, unfortunately for the couple, they have a half percent, yeah, yeah, they have a half, 1 in 2 chances of the children having the same disease as the father. Good job. That was good. And so now, yes. So now, sorry. So now we're gonna talk about the opposite of autosomal dominant. We're gonna talk about autosomal recessive, which means again when we have a complex scientific word, it's always good to break it down. So autosomal, the gene in question is not on a sex chromosome, and then recessive, 2 copies instead are necessary. Okay, so now let's think of a similar example but with a different disease. In this context, we're going to speak about cystic fibrosis, which is a disease that causes a lung issue and buildup of mucus in your lungs, and it's quite deadly. Let's give this context. And I'm telling you that Julie suffers from cystic fibrosis, an autosomal recessive disorder. What is her genotype? Does anyone take a guess? So what we know is that, and also the gene is, let's say that it's governed by the C gene. And so we know that her disease is recessive and that she's affected by it. Hence, her genotype can only be one. Does anyone want to guess?
Mariama says small c, and so does Dan and Abdullah.
Yes. Yes, that is correct. Yeah, that is correct. And I have to change that. I don't know why I put that big C. Sorry, but that is correct. Small c, small c. Yes. And does anybody want to guess Mark's phenotype? And Mark, instead, sorry, is a healthy male.
Mariama says capital C, small c, or capital C, capital C.
Exactly. That is perfectly correct. Sorry for this. Continuing on this, let's think about the same example, but now from the risk of cystic fibrosis. And okay, and so now we have the example of the father, which in the context, let's assume that he was small c. And to simplify, sorry, big C, sorry, I wrote it wrong. Let's think about it. And so what are the possible paternal alleles that we have? Does anybody want to take a guess? So in the context of sperm cells. So I'm saying sperm cells, what allele can sperm cells have? And I'm telling you that the fiber, to simplify things, can only be big C, big C. Any guesses?
Capital C, capital C. Mariama says to repeat the question.
Okay, so yeah, but that was correct. So the question was, if the fiber is capital C, and sorry, by the way, ignore the Huntington. Sorry, I was talking cystic fibrosis, but yes, it would be capital C, because he's a healthy male. So here in paternal allele would be big C, and then B, big C. Now let's talk about the mother. The mother, unfortunately, has the disease. So what would her what's it called? What would her her alleles be if she is small c, small c? What are the possible alleles that the X cell that she carries have? So I'm gonna write this down
Someone
for
says small c, Mariama, and then
perfect.
Abdullah and
Perfect.
Abdullah. Yeah.
That is perfect. So I'm gonna write that on a piece of paper, which I think it's very nice for all do because it can clarify things. So okay, and then maternal, we have small. Can people also see my face, right?
Yes.
Okay, I'm gonna show you some paper as well. So we have here paternal big C, and then maternal small. So that's perfect. Okay, so now let's go through all the possibilities that we can have. So let's talk about mixing. Uh, let's talk about mixing the two people we have. So if we have the big C and a small c, the first mixer we would have would be big and small. So one big C, one small c, like this. Then the same would be for the one below because we still have a big C and a small c. Okay, and and the same goes also for the other side, which would then also be big C, small c, big C, small c. So actually, if we draw out the punishment.
Some people can't see your face. I think because it's quite small there in the
Oh,
background.
okay, yeah. So I did one thing on papers because I wrote this wrong here, and so I will instead. Oh, sorry. Hide. Okay, I will write this in a chat here. Where's the chat? Is on. Yeah. Okay. So what I'm trying to say is, okay, now I can probably see it better. It's probably flipped, which I'm very sorry for. But as you can see, the only possible phenotype we have for all the children is big C, small c. So it's what goes like this. So if all the children are big C, small c, what does this mean about the children? And here we're talking about an uh, yes, exactly. So they are all carriers. So with the concept of carriers, which we're going to explore better later, all it means is that because it's a recessive disorders, they are not affected by the disease. They don't show any symptoms. They don't show any phenotype that is abnormal, but they do have the possibility of passing it on onwards. So if we have this couple coming to our clinic, we would tell them, fortunately, your children are not at risk, but future generations are. And which obviously they would care about. And this is why oftentimes in many recessive disorders, you often have a cyclical nature, where you often have one parent affected, their children not being affected, but the children of the children being affected. So you have this, not always obviously, but oftentimes parents, especially like in older generations where like medicine was not spread widespread, where you would often have the cyclicity of these disorders coming back and forth, back and forth. And okay, sorry, I'll fix the slide because I think it's wrong. And as in it's flipped, but exactly. And as you said before, so the carriers, as yes, and this we're going to explore better later. The concept of carriers, which we just talked about. So this was a lot of information, and I just want to have 5 minutes to take a quick break and to let you guys ask any questions, any concerns, or anything that you want to go through with me before we start talking about the next step. And in the next step, we'll essentially be going through some questions that are going to try and test out your understanding of this. Are there any questions? Okay, there are only okay. And Sainabou, can you okay. So we should not overrun. Okay. So then let's go straight to the questions, which are good. And so if there are any burning questions, we can also do the questions another time. But let's continue. And okay. So then let's test out our understanding of genetic disorders. So now we're looking at another family. I'm sorry if this is a bit repetitive, but it's good to make our understanding much better. Okay, so now we're looking at another family where the father is a carrier and the mother is healthy. Okay, so let's go through this together so we can do it quickly because we only have 15 minutes. The father is a carrier, as the father is a carrier of cystic fibrosis, which we just heard is a recessive disorder. This means that the father has what genotype? Can anybody write this quickly in the chat? So exactly, perfect. So his sperm cells would be big C, small c. Perfect, amazing. And the mother instead is healthy. So if the mother is healthy, any guesses? Exactly. Yes, exactly. So that's supposed to be big C is big C, sorry. But okay, so okay, so continuing on, and I can write this again also on paper. And what are the possible genotypes of the children? Does anybody want to take a guess on what they are?
Mariama says capital C, and Jainaban Laye also says capital C, small c, capital C, capital C.
Yes. Okay. But that is partially correct. Ooh, can you see this? Yes. Perfect. Okay. So I will write this down better later. But yes. So we have because the father is big big small. So the fiber is big small, which means that is correct. But also, we also take into account that as the mother is small, small, they will have also half 102 chances of having two small allele children. So two small C's. What this means is that half the children will be what's it called? Half the children will be carriers, and half the children will not be carriers and be completely unaffected by disease, and also the children. And that's a very key concept to understand, the fact to go back on this idea of cyclicity of genetic disorders. So where we can have them skipping in one generation. And so good job, that is correct. And as I said before here, half of them will be carriers and half of them will not be affected. And okay, now let's talk about another family, and where instead they're both carriers for cystic fibrosis. So if they're both carriers, this means that they both have, as we just talked about now, the big C, small c genotype. So we both had the same genotype. And exactly. What does this mean instead is that we have this instead of genotype in a possible offsprings, where two will be big C's, and sorry, one will be two big C's, two will be one big C, one small c, and one unfortunate child instead will have two recessive cystic fibrosis genes. So if we had this family, what would we tell them about the chances of having a child with cystic fibrosis? So here we see that we have two big.
Is 1 in 4.
Exactly.
As well.
Exactly. So here we would have 1 in 4 of 1 in 4 of having what's it called? Of having cystic fibrosis, which is the small 2 C's. Then 1 in 2 of them being carriers, because we have two examples in which of a big C and a small c. And then we have 1 in 4 again of a possibility of the child being not a carrier and not affected. So we can see here again cyclicity, because we have two people that were not, that are unaffected carriers, but have one that may have a child that is instead affected. So this brings us close to our end. And before leaving, I want to quickly go through the objectives and to see whether we've got an appreciation for for the inheritance of some genetic disorders. And the reason why this is important, because the examples that we went through today, which are recessive and dominant, are the two most common but also simple examples. And in the next sessions, instead, we're going to build on this to understand other types, like, for example, diseases that are linked to sex, to our sex chromosomes. And so before finishing, I want to make sure, are there any questions, burning questions that anyone has? And